Welcome back to Antibody Basics by Biointron, Part 11. In this episode, we'll talk about diagnostics such as ELISA, Western blot, and immunofluorescence. Antibodies are produced quickly in response to infections, making them valuable for diagnostic purposes. They recognize and bind to specific epitopes on antigens. Specificity and high affinity make them ideal for detecting the presence of pathogens, biomarkers, or other molecules indicative of disease. As infections progress, antibodies undergo processes such as affinity maturation and class isotype switching. These changes in antibody distribution also allow for differentiation between acute and chronic infections. They are often used in enzyme-linked immunosorbent assays, ELISA, Western blots, and immunofluorescence. Enzyme-linked immunosorbent assay, or ELISA, is a heterogeneous enzyme immunoassay technique used in clinical analyses. ELISA is a plate-based assay for detecting and quantifying antibodies or antigens in a sample. The target molecule, antigen or antibody, is immobilized on a solid surface. A specific antibody, labeled with an enzyme, is used for detection. The enzyme cleaves a substrate, producing a colored or chemiluminescent signal proportional to the target molecule's concentration. In direct ELISA, the antigen is directly attached to the microplate well, and a labeled primary antibody binds to it. This is straightforward and quick due to few steps, which also reduces the risk of nonspecific binding. However, there are limited sensitivities because of no signal amplification. There are other types of ELISA testing, such as the indirect method. Here, the antigen is immobilized on the well, followed by binding of an unlabeled primary antibody. A labeled secondary antibody then binds to the primary antibody. This format is more sensitive due to signal amplification from the secondary antibody. Various primary antibodies can be used, but it may have a higher background noise. In sandwich ELISA, a capture antibody is immobilized on the well to bind to the target antigen from a sample. A labeled detection antibody then binds to the captured antigen, forming a sandwich. This method is highly sensitive and specific because it involves multiple binding events. It is suitable for complex samples like blood or serum. In competitive ELISA, this is used when sample antigen competes with a labeled antigen for binding to a specific antibody, either attached to the well or in solution. The amount of labeled antigen bound inversely correlates with the concentration of sample antigen. This method can detect small antigens or in samples with low antigen concentration. Another antibody-based diagnostic test is the Western blot. Western blotting is a protein separation and identification technique. Proteins are separated by size using gel electrophoresis. Separated proteins are transferred to a membrane for probing. Specific antibodies, primary and secondary, are used to detect proteins of interest. Detection can be achieved using chemiluminescence, fluorescence, or colorimetric methods. There are several uses for Western blotting, including investigating protein abundance, kinase activity, cellular localization, protein-protein interactions, monitoring post-translational modifications such as cleavage events or phosphorylation, ubiquitinylation, glycosylation, methylation, and sumorylation. Immunofluorescence is a technique that allows visualization of many components in a given tissue or cell type. Fluorescence-based immunoassays offer high sensitivity and real-time detection. Antibodies are conjugated to fluorescent molecules, also known as fluorophores, allowing for sensitive detection of antigen-antibody interactions. After fixation and antigen retrieval, either direct, primary, or indirect, secondary, immunofluorescence is employed. Although the direct method is quicker, the indirect method is more widely used for its high sensitivity, signal amplification, and its ability to detect several targets in the same sample. Multiple detection formats do exist. One includes homogeneous assays, where the antigen and antibody are mixed in solution with a fluorophore that changes properties upon binding. We also have heterogeneous assays, similar to ELISA, but with a fluorescently labeled secondary antibody. In fluorescence resonance energy transfer between donor and acceptor fluorophores upon antigen-antibody binding occurs. The two main methods of IF is direct and indirect. Indirect immunofluorescence, the fluorophore label is conjugated directly to the primary antibody that will be reacting with the target epitope. In indirect IF, a primary antibody binds to the target epitope, then a fluorophore-tagged secondary antibody recognizes and binds to the primary antibody. It is important to understand the applications, strengths, and limitations of your diagnostic test. For ELISA, it is often used for quantification of antibodies or antigens in various samples. It is versatile, scalable, and good for high-throughput screening. However, it provides limited information on protein size or post-translational modifications. The Western blot is used for protein identification, confirmation of expression, size determination, or analysis of modifications. It provides detailed information on protein profiles. However, it is time-consuming and requires gel electrophoresis expertise. Immunofluorescence is used for real-time detection and has high sensitivity and multiplexing capabilities. It is sensitive, rapid, and good for kinetic studies. However, it can be more expensive than ELISA and has potential for background fluorescence. An example of using antibodies as a diagnostic tool is for SARS-CoV-2 serological testing. Commonly used immune-based tests contain SARS-CoV-2 specific recombinant antigens immobilized onto nitrocellulose membranes. Mouse anti-human IgM and IgG antibodies conjugated with colored latex beads are immobilized on conjugate pads. The test sample contacts the membrane and colored antibodies to form latex conjugate complexes. This complex immobilized on the membrane is captured by the SARS-CoV-2 specific recombinant antigen. If SARS-CoV-2 specific IgG or IgM are present in the sample, this leads to a colored band. Another example of using antibodies is through antibody-based proteomics. Personalization of cancer therapy requires the identification of biomarkers for stratification of patients and monitoring of responses to targeted therapies. The generation and validation of specific antibodies offers a high-throughput mechanism for exploration of the proteome and a logical approach for fast-tracking the translation of identified biomarkers. Multiple approaches exist which capitalize on the inherent specificity and sensitivity of antibodies as affinity reagents. Integrating data from high-throughput screening methods, such as DNA microarrays, mass spectrometry and functional genomic screens, with antibody-based proteomics, is an opportunity to identify new cancer biomarkers. Biointron focuses on antibody discovery, expression, and optimization. Our antibody expression service takes your gene sequence to purified antibodies in only 2 weeks. Biointron's high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies, and no antibody means no cost. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca for 1 project commitment, and guarantee the delivery of over 20 unique binders, high diversity, and large capacity. We also have several other services, including but not limited to bispecific antibody production, afucosylated antibody expression, single B-cell screening, hybridoma sequencing, antibody humanization and affinity maturation, recombinant protein expression in the mammalian system, and a CHOK1 staple cell line. Thank you for watching, and comment with any questions or topics you'd like us to cover next. Don't forget to like and subscribe for more.